Good morning, students. Today we are going to discuss very interesting topic, that is the blotting techniques, right? Dear students, am I audible?
Yes, sir.
Very good. Now we are going to discuss about techniques. I mean, blotting techniques, right? Now it is of several types. Before that, what is blotting techniques? Actually, blotting techniques is a technology which is used for specific identification of particular nucleic acid, either DNA or RNA, right? So simply it is to identify particular nucleic acid in the form of either DNA or RNA, or in order to detect or in order to analyze nucleic acid for a specific identification. So that is called as a blotting technique, right? My dear student, blotting techniques is it's a technique which is employed to identify a specific nucleic acid in analytical method, right? That is called as a blotting techniques. So this blotting techniques, it is a broadly divided into following types. The first one is a southern method, the second one is a northern method, and third one is a western method. So there are three methods, right? What is the first method? The first method is a southern method. Second type is northern method. And third one is western method. My dear students, it is very interesting topic. Please concentrate, right? The third type is western. So what is southern method? What is southern blotting? It is broadly divided into following types. They are southern blotting, northern blotting, and western blotting. What is the difference between southern blotting and northern blotting and western blotting? Actually, in southern blotting is nothing but a transfer of DNA from agarose gel to nitrocellulose membrane. That is called a southern method. I like to mention what is southern method. Southern methods are nothing but transfer of DNA. Transfer of DNA to nitrocellulose membrane. So that is called the southern method. Whereas the northern method means the transfer of RNA simply. Instead of DNA, transfer of RNA from gel to nitrocellulose membrane. Whereas western method is nothing but a transfer of protein, particular protein, right? A transfer of protein from electrophoresis to nitrocellulose membrane. So that is called as a western method. So I'd like to mention blotting techniques is simply, blotting techniques is a process which is employed to immobilization of DNA fragments or RNA fragments from gel to nitrocellulose membrane or to blotting paper. So that is called as a blotting techniques, right? On the other one, I can say blotting technique is a tool which is employed for identification of a specific particular nucleic acid, either DNA or RNA, right? So it is also known as, it is an analytical method for identification and the specification of particular nucleic acids in the form of either DNA or RNA, right? So that is called as a blotting technique. Here, blotting technique is broadly divided into three types. The first type is a southern method, southern blotting. Southern blotting is a technique in which a transfer of DNA from agarose gel to nitrocellulose membrane. As a result, we can get a number of copies by hybridization technique. So that is called the southern method. Whereas the northern method is it is a process by which you transfer of RNA from gel to nitrocellulose membrane or blotting paper. That is called the northern method. Whereas the western method is nothing but it is a blotting technique in which a transfer of protein from the electrophoretic layer, right? Electrophoretic process to nitrocellulose membrane. So that is the difference between southern blotting, northern blotting, and the western blotting. Okay, here we are going to discuss about elaborately southern blotting. What is southern blotting? Who discovered southern blotting? And what are the steps to be taken in southern blotting, right? We have to discuss only one topic, right? That is a southern blotting technique. Actually, southern blotting, it is a technique, it was first discovered by Southern, one of the scientists, right? Southern blotting. So it was first discovered by Southern, one of the scientists, very famous scientist, namely, right? He is the man to be discovered southern blotting technique, right? Actually, he's a professor of biochemistry department, right? And he has got a fellowship from Trinity, and was a man to be developed this technique in the year of 1975, right? In addition to that, he has also honored his NESCAR award for clinical medical research for the price of his method of finding a specific DNA, right? So that's why I mentioned southern blotting is a technique which is employed to identify particular nucleic acid, either DNA or RNA, right? That is called southern blotting technique. It is also known as simply, it is a tool which is used for analytical of nucleic acid, either DNA or RNA, right? So that is called as a southern blotting technique. It was first developed by the Professor Sir Edwin Southern, hence it is known as this technique is named by his name, right? That is called southern blotting technique, right? Actually, he has been developed this technique in Edinburgh University, so more than 40 years, right? So Edinburgh University, in which as the man to be created to be developed this technique, right? So hence it is known as a DNA transfer technique or southern blotting techniques. The later on this blotting technique is known as a southern blotting technique, right? Hoping that my dear students you can understand who discovered this technique. Okay, what we are going to see, as he explained, the specific DNA sequence can be developed, right, from the fragments, right? So by using of some chemicals, right, that we are going to say. So simply, the southern blotting is a technique which is going to use for specification or identification of particular nucleic acid, right, DNA. So that is called the southern blotting technique, right? So let me introduce one by one. Actually, southern blotting technique in which the separation of DNA. Now, this is a DNA molecule, right? First of all, the first step, there are so many steps are involved in southern technique. First of all, nucleic acids are taken. First of all, nucleic acids. I think it is a double helical structure of DNA. Now, I like to draw the structure of nucleic acid. You know very well, DNA is a double helical structure. DNA is a double helical structure. Now, there is a double helical structure of DNA. It is to be cut into pieces by using of resection endonuclease, right? Actually, here restriction endonucleus which plays an important role, right? Simply the genome is taken. It is in the form of a double helical structure, which is cut into by using of RE. RE stands for restriction endonucleus. Actually, a restriction endonucleus which cuts a specific site of DNA molecule. As a result, we get now this is a blotting paper. Right, in which by using this section of the nucleus, DNA cut into a number of pieces. So as a result, we can get the fragments of DNA or seen on the blotting paper. So first of all, the fragments of DNA are taken in the blotting paper in southern blotting technique, right? Dear students, you can understand, first of all, a duplicate structure or a double helical structure of DNA is taken, which is cut into piece, a number of fragments by using of an enzyme that is a resection endonuclease, right? It is an enzyme which is used to cut at specific site of DNA molecule. As a result, the helical structure becomes the number of fragments. These fragments are transferred into simply blotting paper, right? And this paper is also transferred into electrophoretic method, right? I'm going to show you the picture that you can understand, right? So this is a picture in which simply the fragments of DNA are taken, right, my dear students? Actually, what is seen the blue in color? They are the fragments of DNA, right? They are the fragments of DNA. Now, as we indicate, it is a DNA. It is the first step, right? And the second step, it is to be left out in a tray containing buffer solution, right? Actually, the second step is we are going to take the second one, beaker is taken. I mean, a tray, plastic tray. The plastic tray is taken in that contains buffer solution by the student. So this region, this is called the buffer. What is the name of the buffer? SSC, sodium saline citrate. SSC stands for simply sodium saline citrate. Now it is used as a buffer solution. Actually, this sodium saline citrate is taken in a tray. This is called a tray. The tray contains sodium saline citrate, which is to act as a buffer solution. That means it maintains the pH value, right? Okay, in that, first of all, this blotting paper containing DNA sequence, right? That is taken inside, right? First of all, before keeping inside the tray containing buffer solution, right? Gel is added. What is added? Gel is added. Here, gel is added. What is the name of the gel? Agarose gel. Actually, what is the role of gel in southern blotting? So simply this gel which separates DNA molecule, right? Simply I can say gel is a solution in which the double helical structure which is converted into fragments of DNA, right? So after leaving this blotting paper into this tray containing buffer solution and just place the one more blotting paper, right? That is called a nitrocellulose paper. So actually this blue color indicates blotting paper. Or simply filter paper. just over the filter paper or blotting paper, we have to place this blotting technique, right? This blotting setup is left out, and just above this blotting paper, we have to cover, we have to leave the yellow color, that is a nitrocellulose. Actually, nitrocellulose. It is one of the membrane, right? Nitrocellulose membrane is placed just over this blotting paper, and just above the nitrocellulose paper, we have to cover, we have to place a number of blotting paper or tissue paper. Simply, I can say one more. This is called a filter paper. More number of filter paper are placed just over the nitrocellulose membrane. So as a result, what will happen is, after keeping this nitrocellulose paper just above the nitrocellulose paper, by keeping filter paper, a number of filter paper are arranged one above another and which are left out just for the nitrocellulose paper. And after that, we have to keep some weight. So which is made for simply glass plate it is also left out. This is a weight, right? So leave the setup for undisturbed place for few hours. After sometimes, what will happen is, so this gel which starts moving from one place to another place. Actually, gel is there where the gel is present, gel is found in the region, right? Actually, this gel which starts, I mean, this sodium cyanide citrate which starts moving. I mean, it is with from the lower paper to just to move towards upper portion. I mean, which is covered with a nitrocellulose membrane paper. Actually, this yellow color indicates a nitrocellulose membrane paper that gets simply the gel from the bottom paper. You can see, actually, there are two blotting papers are inserted. One is the bottom side, one paper is present at the bottom, and at the middle you find this blotting technique, blotting paper, and just above the blotting paper you find a nitrocellulose membrane, and just above nitrocellulose membrane you find a number of tissue paper are arranged one above another. Okay, so when the buffer solution starts moving from one place to another place, as a result, so simply the fragments of DNA are fixed on the nitrocellulose membrane paper. When the buffer solution moves from one place to another place, we can find, after sometimes, right, my dear students, after sometimes what will happen is the DNA fragments are present in the blotting paper which are fixed on the membrane or the nitrocellulose paper. My dear student, please understand, don't get confused, right? Actually, this yellow color stands for nitrocellulose membrane. Nitrocellulose membrane, right? And the blue color is the blotting paper, right? Or filter paper, or this blotting. So this blotting paper that contains simply fragments of DNA. So when the buffer solution moves from a tray into tissue paper, from the tissue paper into gel, the buffer solution which reaches finally on the nitrocellulose membrane. As a result, the membrane, the surface, the bottom surface of the membrane is fixed with some amount of DNA. The fragments of DNA that we have taken in the tissue paper, which are to be copied, upper surface of the nitrocellulose membrane, which is yellow in color, right? On seeing the diagram, you can understand, my dear student, right? So as a result, the membrane, the fragments of DNA are copied or fixed on the surface of nitrocellulose membrane, which is yellow in color, which you see in this picture, right? Okay. And next, this after removal of this membrane paper, it is to be left out. It is to be kept in a hybridization bottle, right? One the bottle is taken. They're going to show you, right? You can understand. Now this is a Hybridization bottle, it is a cylindrical in shape, right? So when it is a transfer, this like a membranous paper which contains a number of fragments of DNA. Simply it is to be transferred, right? In the step, it is to be transferred. What is transferred? The yellow color membrane paper, it is to be transferred. My dear student, please understand and try to follow me. The yellow color paper or the membrane paper or nitrocellulose membrane which consists of fragments of DNA which are left out in hybridization bottle. Now this is called a hybridization bottle. After leaving inside, it is to be rotated for a few minutes, right? So because there is a probe is there, hybridization probe is there, and that is to be rotated for a few minutes. After sometimes what will happen is the fragments of DNA are separated along with the hybridization probe, right? So as a result, we can get the separation of DNA instead of the hybridization bottle. So now this is a process to be hybridization, right? Here it is to be hybridization. Hybridization is a process by which the copying of nucleic acid in the form of either DNA or RNA, right? So that is called hybridization, exact copying of DNA, right? From the nitrocellulose paper into hybridization bottle that contains hybridization probe is there. Now this probe, when it rotates, automatically the fragments of DNA are separated, right? And one more thing, we have to expose to autoradiography. One more instrument that we have to use, that is autoradiography. My dear student, please understand. Here one of the instrument that we are going to use, that is autoradiography. What is autoradiography? Autoradiography that detects the presence of nucleic acid, exact copying of nucleic acid that can be detected, right? So when we use for autoradiography, one of the instruments, autoradiography. It is an instrument which is used to do the detection of some nucleic acid which are present in the nitrocellulose membrane. So as a result, it is to be copied from the nitrocellulose membrane into another blotting paper, right? Here we got to take another blotting paper. So as a result, just we can detect a number of DNA fragments from hybridization bottle when it is used, when it is exposed towards autoradiography. What is autoradiography? It is an instrument that emits continuous flow of light, right? So when the light passes from the bottle, I mean hybridization bottle to another tray, so simply by passing of emulsive light, so the autoradiography which produces continuous flow of light. So the light which will capture the fragments of DNA when it is exposed to autoradiography, as a result, the image of DNA is formed on the screen. I mean, in the blotting paper, right? So this is called simply southern blotting technique, right? My dear student, please understand there are several skills there. The first one is simply giving a structure to fragments by using a prosection window nucleus. The fragments of DNA are transferred into blotting paper and which is left out in beaker containing, I mean, a tray containing buffer solution. That solution is called the sodium saline citrate. It is to act as a buffer because buffer is a solution that maintains its pH value, right? and just to over the sodium saline citrate to find. My dear students, kindly cooperate. Please mute your mic, please. So as a result, the solution which starts moving from one place to another place, first of all, the tissue paper which you absorbs this gel, and the gel has to be drawn off to the top membrane, top side of the nitrocellulose membrane. So as a result, the DNA fragments are fixed on the surface of, the surface of nitrocellulose membrane. As a result, here we can see the third picture in which you can see a nitrocellulose membrane is fixed. When we separate the membrane from the blotting paper, we can find the copying of DNA fragments, right, which are fixed at them. Now it is to be transferred into hybridization bottle. After transferring hybridization bottle, it is to be rotated with the help of hybridization probe, right? After some time, what will happen is some fragments are separated, and after it is to be exposed to autoradiography. So when it is a transfer to autoradiography, we can find simply a transfer and the image which is formed on the screen. Exactly this copied from the nitrocellulose membrane. As a result, exactly the DNA fragments are detected as it is in hybridization bottle, right? So this is called the Southern blotting technique, right? It was first discovered by Southern, right, in the year of 1975. Hence, it gave its name to the technique. Later on, it is known as the Southern blotting technique. Simply, I can say Southern blotting technique is nothing but the transfer of DNA from aqueous gel to nitrocellulose membrane. As a result, we can get its copying. Exactly the detection of DNA fragments from the DNA model, right? So this is a sample to be detected in large amount, right? That is called the Southern blotting technique. Sometimes it's going to be asked in your examination as a 5-mark question, right? My dear student, please understand, explain or write about the steps to be involved in Southern blotting technique. If it is asked, you got right? What is Southern blotting technique? Transfer of DNA from gel to nitrocellulose membrane. What are the steps to be taken then? First of all, DNA cuts, the fragments are transferred. A transferred DNA are left out over the tissue paper. Just over the tissue paper, place the nitrocellulose membrane, and from the nitrocellulose membrane, it is to be removed and it is to be transferred to hybridization bottle. Then expose this hybridization bottle after rotation into autoradiography. As a result, the image and formed from the nitrocellulose membrane, the spray, which are showing resemblance like as it is from the hybridization bottle, right? So simply the detection of DNA. Here, DNA is detected by the technique that is called the Southern blotting technique. Yes, students, did you understand? First of all, my dear students, did you understand? Students, am I audible?
Yes, sir.
Yes, sir.
Could you mention the name of the enzyme which plays an important role in Southern blotting? The first one. Will you mention? One of the
Restriction enzyme.
very good.
Restriction enzyme.
Restriction enzyme. What is its role simply? What is its role?
It leaves the DNA fragments.
It cuts DNA molecule, right? Okay. In first diagram, you see the pictures blue in color. What is it exactly? They are the fragments.
DNA fragments.
Very good. DNA fragments. Okay. Will you mention the name of the buffer solution which is present in the tray?
SSC.
SSC. Could you abbreviate, please? What is the abbreviation?
Sodium salicylate.
Sodium saline citrate.
Great. Very good. Actually, it is used as a buffer medium, right? Okay. In the bottom, you see one of the paper. Could you name of the paper which is yellow in color?
Autoradiography.
Here, okay, autoradiography is involved in that. Okay, no problem.
Blotting or filter paper.
Blotting paper, very good one. So one more gel is there. One more gel, right? What is the role of gel? Here, you mentioned the role of gel.
Andros gel.
Agarose gel. Actually, while it is kept out in agarose gel, it is going to be dissolved. I mean, it is to denature. I mean, the 3D structure of DNA is to be lost when we transfer into gel. So that is the role of agarose gel. Dear students, denaturation, how it is taking place? By applying of one of the gel. When we place DNA into agarose gel, what will happen? DNA gets denaturation. What do you understand about denaturation? Simply, its original structure is lost, right, students? Okay, and one more bottle is there, that is hybridization bottle, right? And finally, what is to be deducted? DNA. My dear students, actually, who discovered this technique?
Edwin.
Edwin Southern.
Very good. Very good. Keep it off, right? Here, sometimes you can expect a very big question, that is write about the steps involved in southern blotting technique, right? If it is asked to just write its definition, then draw the structures, right? And just explain one by one, right? Which is very important area. Next, I'm going to compare. Already, as I have discussed a number of techniques like southern blotting, northern blotting, and western blotting. Now, I'm going to compare the comparison between each technique. Okay, my dear students. Next, I'm going to compare. So this is also very important area, as I have discussed a number of techniques, right? They are southern blotting and northern blotting and western blotting, right, my dear students? So the first blotting is southern blotting. So there are some processes taking place. The first one is a separation of DNA. And second one is the denaturation. It is to be lost, denaturation. And third one, type of membrane is used, right? And fourth one is hybridization technique. And fifth one, next one is visualizing, right? How can we see? That is a visualizing, right, my dear students? This is a technique that I'm going to compare. So the first of all, I'm going to show you, right, my dear students, separation of DNA, right? Actually, the separation of DNA is simply in southern blotting, a DNA is separated, whereas in a northern blotting, RNA is separated. I mean, the transfer of RNA from DNA, right? That is called the northern blotting, or the transfer of protein from agarose or electrophoretic process to the nitrocellulose membrane, that is called the protein. Simply, what is separated in the first step, that is DNA, and whereas in southern blotting, northern blotting is RNA, whereas in western blotting, protein is separated. Second point, denaturation. Denaturation means the original structure is lost. When we place an agarose gel, right? So that is called denaturation. Simply, when we dip DNA into gel, what will happen? The double helical structure, it is going to be lost. So that is called the denaturation. Here, denaturation is needed. My dear student, denaturation is needed, whereas in our northern blotting it is not needed. Without that we can transfer easily RNA to nitrocellulose membrane. And talking about western blotting, this denaturation is highly needed, right? It depends. Next one, the type of membrane which we are using in southern blotting and the northern blotting. So here there are two different nitrocellulose membrane are used. The first one is a nitrocellulose membrane. It is a blotting membrane that is used for copying of DNA fragments in case of southern blotting, whereas in northern blotting, one of the membrane that is widely used, that is amino benzyl oxymethyl or methyl. Amino benzyl oxymethyl, right? This is a membrane which is used in northern blotting. Here, my dear student, you can expect which of the following membrane is widely used in northern blotting. If it is asked, you have to mention A, nylon, B, nitrocellulose, C, amino benzyl oxymethyl, right? Next, talking about western blotting, same nitrocellulose membrane is also used. And hybridization, actually from DNA to DNA, that is hybridized. I mean, DNA is a double helical structure. From the structure, it is again DNA is to be detected, right, by southern blotting. Whereas in northern blotting, RNA is taken from RNA, DNA is going to be detected, right? DNA is going to be produced or made, right, by the process of hybridization. Whereas in western blotting, a protein is hybridized by the process, right? Whereas visualizing, right, visualizing that is used, visualizing is made possible by using of autoradiograph or autoradiography or autoradiogram. Visualizing that is applied autoradiogram, whereas in northern blotting, the same autoradiography is also applied. Whereas in western blotting, it is a dark room is essential for detection of proteins. So these are all the comparison between southern blotting, northern blotting, and western blotting. My dear students, it is very important to write the comparison between blotting techniques. If it is asked, just make a tabulation. The first one is a separation, second one is a denaturation, third one is a membrane, and fourth one is a hybridization, and next one is a visualizing, right? So now I am going to give you home assignment, and all of you take your home assignment and try to read all the questions thoroughly, right? And try to practice yourself by writing in your note, right? Thank you for joining, my dear student, and please take your home assignment. The first question, what is blotting technique? What is blotting technique? Second one. What is southern blotting? And compare or differentiate. Northern blotting from western blotting. And fourth one. Write the differences. Between blotting techniques. Last one more question, very interesting question, that is explain the steps involved in sudden blotting technique. Explain the steps involved in sudden blotting. Right. Thank you for joining, my dear student. Try to take down all the questions and read thoroughly. Practice thoroughly, right? In examination point of view, definitely you can expect 1 more question, 2 more question, 3 more question, and 5 more question. Thank you.